大家好，欢迎大家抽出周五的宝贵时间来参加我们美墨尔特2022年第一季度的网络研讨会。那么今天我们网络研讨会的主题是 ICH Q1A R2 稳定性测试。我的名字叫做李媛媛，来自中国，是负责美墨尔特部分亚洲区域的区域经理，协调中国和印度的子公司，以及在韩国、新加坡和马来西亚的区域销售。那么今天在现场陪伴我的呢是来自市场部的安妮卡，她负责录影跟摄像。在对话框中有我们美墨尔特上海的产品经理 Orange， 他会在对话框中对您所有的疑问进行解答。请您放心，我们的对话框都是加密的。如果对于此研讨会中有任何的疑问，欢迎告知我们。那么今天主要介绍的是稳定性测试。以前是没有稳定性这个概念的，一系列事故促使监管方重视产品的质量和安全问题。那什么是 ICH？ 在 ICH 指南的要求下，什么又是稳定性测试？适用于什么区域？接下来的30分钟，这些问题都会在我们这次的网络研讨会中一一被解答。我先会跟您做一个简短的介绍，然后会请出我们美墨尔特专家 Andrew Weiss 用视频向您进行更深入的探讨个别测试研究的类型和参数细节。最后我会向您展示完美匹配稳定性测试的美墨尔特专有解决方案 HPP ECHO 系列。那什么是稳定性测试呢？药品在投放市场之前呢，需要做一系列的测试，经过审批之后呢，才能达到销售许可。在这一系列测试中，很重要的一环就是稳定性测试。稳定性这个概念呢，来源于可靠性。可靠性评估呢，多应用于工业领域，其十分常规成熟。那么稳定性的范围界定于化学稳定性、物理稳定性以及微生物稳定性。严格的验证测试是在产品和包装上进行的，如果不是十分安全，就会对人体造成巨大的伤害。在医药行业中，药品的产品和包装在上市之前会经历一系列的严格的考察，例如对疫苗，而其他非制药行业也可参照 ICH 指南进行质量检测，如在化妆品行业的个人护理，针对产品和包装，或者是针对科学领域的研究，以及在食品行业，例如确定保质期限等等。那什么是 ICH 指南呢？ICH 指南，国际人用药品注册技术要求协调会撰写了医药产品稳定性实验要求和指导。ICH 成立于1990年，旨在协调人用医药产品评价中技术指南的解释和应用，作为药品的审批的基础，以尽量减少开发和审批程序中的重复。这个组织是一个将欧洲、日本、美国监管行业以及制药部门的专家聚集在一起，讨论药品注册的技术和科学的项目。2021年6月 ICH 第一次大会上，中国 NMPA 国家药品监管总局连任 ICH 管理委员会委员。国内 NMPA 将继续参与 ICH 国际事务 ，ICH 的一些指导原则也将逐渐转化为国内的业内标准或者是技术规范。我们也注意到，国内的NMPA 这几年密集发布了一系列技术指导原则，这就需要我们对 ICH 有一个更深入的了解。那么ICH 指导原则的实施推动了技术要求与国际接轨，跨国企业可以更早地将早期的研发项目引入中国，同步研发、同步注册已逐渐成为可能。ICH Q1A R2 的摘要全面讨论了行业支持和实践的现行法规，在批次选择、包装、质量标准、检测频率、储存条件、稳定性承诺、评估和标签等几个方面，对如何开展原料药与药品的稳定性研究提供了指导。 那 Q1B 呢，给光稳定性实验提供指导。新原料药和新药制剂应经过适当的光稳定性特征考察，证明其本身的光稳定性。那么现在我很荣幸，请安妮卡给我们播放下来的视频。我们的专家高级区域经理 Andrew Weiss 会给您进行更详细的介绍。Andrew Weiss 在美墨尔特已拥有超过15年的工作经验，目前呢是负责在东南亚区域的销售管理。由于他在 ICH 领域的多年研究，已经在全世界各地为不同客户进行了培训和指导。视频配有中文字幕，请欣赏。Annika, please.
Stability testing in accordance to the quality guideline ICH Q1A, which is the second revision, emphasizes the stability testing criteria to be fulfilled for successful stability testing. Thereby, it is requested to issue the comprehensive documentation data needed for the registration application of the new drug substances and products. Documentation package, this includes, for example, the stability test protocol. There will be given information on the batches which are tested, container closure system, storage condition, storage period, testing specification. Also, they have to be issued a stability report with details on the defined analytical methods, the results achieved, along with evaluation conclusions, statements, and the summary. Let's go further to stability testing. Stability testing is a complex process which involves factors influencing the stability of the product, which consequently are affecting the quality of the product. Consider active ingredient stability, the interaction of active ingredients and excipients, the formulation process, which is the mixture, the dosage form, the manufacturing, and the type of packaging selected. All these parameters are important for the stability of the product. Acceptable stability requests the product to remain within its specified limits throughout the period of storage and use, which means the same properties and characteristics as possessed at the time of packaging shall be kept until its consumption. Here we talk about the physical properties which must be remain. That is in order to avoid physical change, which influences the biological availability. Then there's the chemical properties. This should be kept as chemical change may lead very often to higher amount of degradation. And microbiological properties, these are important as exceeding the maximum number of acceptable terms. This is harmful and dangerous for the patients. There are guidelines for approval and acceptance which are going to be followed. This is ICH guideline or FDA guidance and GMP regulations. And stability testing, this evaluates the effect of environmental factors on the quality of the product to predict a shelf life, determine proper storage conditions, and suggests labeling instructions for the product. Drug development process. This starts with the excipients, which is a substance formulated alongside the active ingredients. This is also called bulking agents or fillers. This could be lactose or paraffin, water, ethanol. All this is used as a thickener of soft capsules or talc. Then we have the new drug substance, which is the active product ingredient, and this has to be tested for stability and impurity. Then we get the formulation development. This is the composition of the drug substance and the excipients. And then this has also to be tested in regard to stability, excipients, compatibility, formulation, interaction, and additional to the testing of this finished product, also packaging is very important for the stability testing and this has to be considered. So we have here primary packaging that could be ampoules, vials, containers, blister packaging. That is the one it is in direct contact with the dosage form. And we have the secondary packaging. This is the packaging which is used as an additional layer of packaging, normally not requested for hygiene, preservation or protection of the drug itself, and it will not be in direct contact with the drug. It is important to determine and assure the identity, potency and purity of the ingredients as well as those of formulated products. ICH, this is an abbreviation, and that is International Council of Harmonization of Technical Requirements for Pharmaceuticals for Human Use. This is a global initiative, and the organization has about 17 members and 32 observers. It wants to bring together regulatory authorities and pharmaceutical industry to discuss scientific and technical aspects for pharmaceuticals. Develop also ICH guidelines and achieve greater harmonization worldwide to ensure that products are safe and effective and in a high quality. And determine stability data package for new drug substances and drug products, which are sufficient for the registration application within the three regions of European Community, Japan and United States, which refers to the climate zone 1 and 2. Stability testing is used in order to establish how the product changes over time under environmental factors such as temperature, humidity, and light. For drug substances, stability testing determines the retest date, that is the time frame during which the drug substance is expected to remain within its specification and therefore can be used in the manufacturing of the drug products. And it helps to establish data to support setting API specification and data to support submission of drug product. And for the drug products, stability testing is necessary to fix the shelf life of the product. This is the duration for which the product is safe to be used and retain within the therapeutic effect. And also it helps to establish recommended storage condition and to issue the data to support setting specification for the drug product. Coming now further to the testing. There is the first one, which is the stress testing, which is the force degradation study. The force degradation study is an exposure of the drug substance or drug product to different stress conditions, which results in relevant degradation products. During the pre-formulation, it supports the selection of the compounds and excipients for the further development. And for products, it provides information on the degradation mechanism and the potential degradation necessary for the manufacturing process and also for the packaging. The guideline requires conducting force degradation studies under a variety of conditions as pH, light, oxidation, dry heat separation. So there's mentioned should examine the effects of temperature in 10 degrees increment, e.g. 50 degrees, 60 degrees above accelerated testing. The humidity to be examined at 75% RH or greater. It should be carried out on a single batch of the drug substance. Testing in solution should also be performed across a wide pH range, either solution or suspension. And also photostability testing is an integral part of stress testing. That is the testing along with the light. Important here is always to get primary degradation and not completely degrade API. And when the objective of stability study is stipulated and the method is certain, the drug will now be evaluated under a much larger scale, and now stability study is performed. And here we are talking about the long-term studies, accelerated studies, and intermediate studies. Long-term testing, that is stability studies under recommended storage condition for retest period or shelf life, proposed or approved for labeling. That is the real-time testing. Or the accelerated testing, studies designed to increase the rate of chemical degradation or physical change of a drug substance or a drug product by using accelerated storage condition. It is necessary to do long-term testing and accelerated testing. That is a must. And then there comes also intermediate testing into effect in case accelerated testing cannot successfully be done. Then the starting is here at 30 and 65%. That is provided on the fact if long-term testing is done at 25 degrees and 60% RH. Let's go further to the storage testing. Here we can see the general case which applies if the drug substance is not covered by a subsequent sections. Here there is stated that long-term testing has to be done at 25 degrees 60% or 30 degrees 65%. The minimum time period covered by data at submission should be 12 months, and it must be in a limit of plus minus 2 degrees and plus minus 5% RH. At least 3 primary patches at the time of submission will be tested, and the testing is every 3 months first year, and then after registration process you have to go on, of course, with testing. Then it means every 6 months the second year, and annually thereafter continue studying to cover the full storage and shipment time. The accelerated testing, that is done as 40 degrees 75%. This is 6 months for the registration purposes. And here we also have remarks of plus minus 2 degrees and plus minus 5% RH. And the intermediate testing, which comes into effect if accelerated test fails and long-term testing is done at 65 degrees. And that means we have here the remarks also plus minus 2. And it is done at 30 degrees and 65% RH. Let's go further to the storage in refrigerator. This means products intended for refrigerated storing, for example, vaccines, which might be stored under refrigerated conditions, or DNA oligonucleotides. Here, long-term testing is at 5 degrees for 12 months, and here we are in a limit of plus minus 3 degrees. And the accelerated testing here has to be done at 25 degrees and 60% RH. And this is also for 6 months, and limit is plus minus 2 degrees and plus minus 5% RH. Storage in a freezer. This is actually very rare. Here the testing has to be done at minus 20 degrees for 12 months in a limit of plus minus 5 degrees. In the absence of accelerated storage, the testing of single batch at elevated temperature, for example 5 degrees plus minus 3% or 25 degrees plus minus 2 degrees for appropriate time period to address short effect term excursions outside proposed laboratory storage condition is requested, and here that is for shipping and handling. And there's storage below minus 20 degrees. That is case by case, is very seldom. That is also for vaccines. So here, for example, could also be lower than this minus 20 degrees. Then we have the testing in regard to the impermeable and semi-permeable containers. That is the sensitivity to moisture or potential for solvent loss. That is here the concern, and that means if the product is packed in impermeable containers, the testing can be done under any controlled ambient humidity conditions. If we go for the semi-permeable containers, that means it allows some material to pass through, but disallows other material to pass, and that is aqueous-based products which are packed in semi-permeable containers. This should also be additionally to physical, chemical, biological and microbiological stability evaluated for the potential water loss and this can be done under lower humidity. And here the customer can decide if to do it at 25 degrees 40% RH or 30 degrees 35% RH. Alternatively, starting at higher humidity is possible, that is deriving the water loss at the reference relative humidity through calculation. Here accelerated testing is also mentioned, that is 40 degrees 25%, or also intermediate testing in case accelerated testing fails. ICH guideline covers climate zone 1 and 2. Here on this chart we can see the different climate zones available on the globe, and here we have also mentioned WHO, and there is referring to climate zone 3, 4A and 4B. Climate zone 3, this is climate hot and dry, and here the testing shall be done under 30 degrees and 35% RH. For the climate zone 4A, which is hot and humid climate, the testing is for 30 degrees and 65%. And for climate zone 4B, the long-term testing shall be done at 30 degrees and 75% RH. So that means that there's a coverage of the global zones worldwide. And we have here the worst case, which means for 1 and 2 climate zone, testing 25-60. For climate zone 3 and 4A, 30-65%. And climate zone 4B, 30-75% RH. All these parameters are covered by the MEMAT climate chambers. Our constant climate chambers, HPP Echo, which is mostly efficient for the use of long-term and accelerated testing under all these climate conditions. Or our climate chamber ICHL, which can be used for the photostability testing. Please kindly get now more details and information from my colleagues, who is introducing to you the most important features of our MEMAT climate chambers, mostly useful to successfully perform the manifold stability testing.
欢迎回来。SHQEA R2 1.3 的通用原则呢，指出稳定性测试的目的是根据所提供必须的证据，证明在多种环境因素，例如温度、湿度和光线的影响下，原料药或者药品的质量如何随时间变化，以及确定原料药的重新测试期限和药品的有效货架期以及建议的储存条件。药品虽然可能在一个地区生产，但是使用和销售会可能遍布全球各个地方。当然，你已经从我们的视频中专家那里了解到，稳定性实验研究包含的长期实验、加速实验以及中间实验的考察条件。从气候资料获得的平均动力学温度，世界可分为4个气候带。一般条件储存的药品，在长期实验中温度和湿度的选择，通常依所处气候带的条件而定。中国总体来说处于亚热带，部分地区属于湿热带，跨越2区与4区。2020年版中国药典9001原料药物与制剂稳定性实验指导原则，故长期实验采用温度为25度加减2摄氏度，相对湿度为60%加减5%，或温度30摄氏度加减2摄氏度，相对湿度65%加减5%，与 SH 采用的条件基本是一致的。另外 ，FDA 在联邦法规第21章第203条要求，药品制造商、授权经销商以及代理商应当在能够保证其稳定性、完好性和有效性的条件下，储藏和处置所有药品的试样，避免药物试样受污、变质和掺假。环境测试箱体可模拟设定的环境参数，必须接受控制、监控并进行存档。 为了降低分布研究失败的风险呢，要求使用设计上满足功能和合规性要求的监测系统。这就是美墨尔特产品的用武之地。我们的环境测试箱可完美的模拟稳定性测试所需要的所有参数，是您的不二选择。配备的 Atmo Control FDA 系统实时监控箱内的温度、湿度、光照、紫外等参数，并对数据进行编辑记录和评估，保证包括电话或短信报警提示在内的连接选项，包括数字签名、完整事件和交互历史以及审计跟踪在内的多层次数据安全性。带有帕尔贴半导体技术的 HPP Echo 专为在受控条件下进行长期测试而做准备，并具有其均匀且稳定的温度和湿度的分布。参数的精准控制满足您对 ICH Q1A 稳定性测试的极高要求。当然，另一款恒温恒湿箱 ICH Echo 气套式制冷和加热系统包围在整个内腔内，确保快速和精准的温度控制。其无与伦比的温度和湿度的均一性非常适合长期的稳定性研究，满足 ICH Q1A 的要求。带有光照模块配备的冷白灯或者是紫外灯 ICH-L 以及 ICH Echo L 符合 ICH Q1B 的光稳定性测试要求，也满足符合全球化妆品以及食品的稳定性测试的标准。那我们在现场呢，给大家展示了一款我们的 HPP410 Echo。那HPP Echo 系列呢，采用了先进的半导体技术，秉承美墨尔特社会责任的理念，运行安静、环保、节能，并无噪音。美墨尔特作为第一个将此技术推广应用到温控设备领域的行业佼佼者，精准控制参数呢，是我们对客户的承诺。在连续运作时，它比压缩机冷却设备能源节约效率高达90%。不需要任何制冷剂，也无需特殊的维护。HPP 的温度控制呢，是从0度到70摄氏度，以及从10%到90%的相对湿度进行主动加湿和除湿。这些都是根据制药业稳定性测试的要求 Q1A 进行优化设计的。那么我们有7种型号可供您的选择，从容积108至2140升，满足不同阶段的测试需求。当然，温度控制系统卓越，采用纯化水或者蒸馏水，没有循环水，也没有废水排放，换水周期长，一般3到6周，维护比较简便。采用直观方便操作的Twin Display 2块显示屏，操作简单，精准控制温度、湿度以及计时器，还有报警。当然，也可以对数据进行编程，包括能观看数据的运行情况。此外，内外采取独有的美墨尔特不锈钢，方便清洁和维护，没有死角。我们看一下内部。内部配有玻璃门，可以更方便对内部箱体进行观察，避免外部的污染进入箱体内部。同样的话，可以避免温度的陡然上升和下降对测试环境进行影响。我们看一下内部，相比于上一代的HPP Echo HPP410来说，上一代的 HPP410配备了5块半导体模块，而如今升级版的 HPP Echo 410呢，配备了2块先进的半导体模块，优化了控制温度和湿度的精准程度。此外，我们也可以看到，在这里呢，配备了两个PT100的温度传感器，还有这里的湿度传感器。从这里也能看到，这两边是2个除湿的半导体模块，它强化了内部的除湿效果，消除了内部结冰的隐患。 我们再往后面看一下。从背部我们可以看到，这里配备了以太网的接口以及电源的插口。这边是2块半导体模块进行加热加热还有冷却的背面，包括2块除湿的半导体模块，以及包括配有的冷凝水盘。那冷凝水盘呢，可以进行拆卸，方便维护。那我们也看到这里呢，是配备了先进的和优化性的蠕动泵。那么蠕动泵呢，优化了我们的湿度控制系统，包括呢更容易方便的清洗和维护。所以这是我们在升级了HPP Echo 的优点。最后回归主题，今天呢，我向大家介绍了 Q1A 关于稳定性实验的一些概念法规。同样，我们美墨尔特的针对稳定性测试的完美的解决方案，也是我们产品行列中非常重要的，也非常受市场欢迎的 HPP Echo 系列。当然，如果以后有机会，我会更乐意跟大家介绍不同领域的其他法规，以及我们的其他产品，如 SAH 以及 SH Echo。希望这次研讨会使你们受益。内容也很丰富。安妮卡呢会事后将Wise 女士的 PPT 上传到网页上，供您方便下载和更深入了解。如果有任何的问题和反馈，包括对我们整个研讨会有任何的意见和见解，欢迎致电我们，欢迎联系您在美墨尔特的专属联系人，或者发邮件到 webinar@mehmet.com。十分感谢您抽出这30分钟的宝贵时间，与我们共同认识了稳定性测试。也感谢我的团队对我们这次研讨会的全力支持。那十分感谢，我们下次再见，拜拜。